Qkine is a primary manufacturer of growth factors and other complex proteins. The company was established in 2016 as a spin-out from the laboratory of our CSO, Margot Hoovenen, at the University of Cambridge. Our ISO 9001-2015 certified manufacture and R&D facility is located on the Cambridge Science Park in the UK. We manufacture our products using a highly defined microbial production process to provide high-purity, entirely animal-free growth factors. Our R&D is focused on developing new production processes and addressing specific customer challenges across a range of stem cell culture applications. The company was established by two scientists, and our principle is to provide the highest purity bioactive proteins to best support our customers and maximize the impact of their science. We produce our products using microbial expression systems, and each step of the process is assessed and certified as animal-derived component-free. This ensures there is no possibility of contamination by closely related endogenous mammalian growth factors, which is an issue when using a mammalian protein expression host, or indeed by animal contaminants from the production process itself. True animal-free growth factors, such as ours, are used in chemically defined cell culture media to facilitate the scale-up and translation of stem cell-based systems for clinical or drug discovery applications. Considering the source and quality of essential reagents, such as growth factors at an early stage, even during basic research and in subsequent process development, helps support efficient and effective translation and the end goals of our customers. Until recently, no fully animal-free TGF-beta1 proteins were available. This was problematic for the industry, as TGF-beta1 is a core component of the base chemically defined stem cell media E8. Our team, building on decades of experience with TGF-beta family growth factors, have created a production process that allows this complex, disulfide-rich protein to be produced in a microbial system and formed the correct tertiary protein structure, which is important for maximum bioactivity and stability. We are the only supplier of TGF-beta1+, which has exceptional lot-to-lot consistency and a demonstrable advantage in iPSC culture systems. In these data, TGF-beta1+ has been used in combination with our FGF2, also known as basic FGF, for maintenance of pluripotency. The efficacy is shown by the highly homogeneous immunofluorescence staining with the pluripotency marker NANOG. At Qkine, we are passionate about solving customer challenges. Those stem cell biologists working in the important area of liver regeneration express frustration at the lack of animal-free growth factors for hepatocyte, or liver cell, differentiation from iPSC. HGF is a complex growth factor, not amenable to animal-free manufacture. However, we have produced the naturally occurring NK1 isoform and shown the successful generation of hepatocyte-like cells. Indeed, our HGF-NK1 was shown by this researcher to give a more homogeneous hepatocyte-like cell population at a lower concentration of 10 nanograms per ml than the mammalian cell-expressed control. The addition of animal-free oncostatin M and HGF-NK1 completes the set of growth factors we provide for the hepatocyte differentiation workflow. Cells in their physiological niche in the organism are subject to a complex interplay of physical and biochemical signals. When any cell population is removed from this environment and placed in culture, it is primarily the growth factors in the media that substitute for these signals to control cell proliferation and fate. However, this is expecting a lot from isolated proteins that have also been removed from their niche and the myriad of interactions that modulate their activity and behavior. One of the most obvious of these is the intrinsic instability of some proteins in this artificial milieu. Of these, FGF2, which is essential in most pluripotent stem cell culture protocols, has a functional half-life of less than 1 day. In practice, this means that as soon as the culture media is changed, the effective concentration of the FGF2 starts to reduce in that culture, leading to heterogeneity and the time-consuming and expensive requirement for frequent media changes. Thermostable FGF2, which is the best technology to date that solves this challenge, was developed by Masaryk University and the biotech company Enantis. Known as FGF2G3, this has already been used in new weekend-free medias in basic research applications. We have licensed this patented technology and combined it with our manufacture expertise to make available for the first time a tag-free form, which is more compatible with translation and scale-up, and removes scientific uncertainties due to the presence of his tag. You can see from these data that at day 0, when the media is first changed, the potency of the two forms of FGF2 is equivalent. However, by day 2, the activity of the standard FGF2 shown by the blue line has dropped considerably and is barely detectable after 7 days. We can't present you with all our products today, but we wanted to highlight briefly some of the emerging areas that we are focusing on where we are using our core expertise in protein engineering and manufacture to address the next set of challenges for cell biologists in areas such as cellular agriculture, bioprinting, tunable enhanced functionality synthetic hydrogels, and unmet needs in the organoid sector. Qkine was founded on strong scientific principles. Every batch of protein manufactured undergoes extensive biochemical purity analysis and functional bioactivity testing using a quantitative cell-based assay. Endotoxin contaminants can be a concern for microbial expression systems. However, by following rigorous processes, we achieve nearly undetectable levels of endotoxins and have set industry-leading low endotoxin level acceptance criteria for our proteins. Certificates of analysis, animal-free certificates, MSDSs, and certificates of origin are available for all Qkine products, and we are happy to consider additional bespoke testing if this is required for your application. To conclude, I'd like to highlight why such stringent quality is becoming ever more important and is a founding principle at Qkine. Organoids are stem cell-derived 3D culture systems that can now recreate the architecture, physiology, and disease states of human organs and tissues in remarkable detail. Organoids are often grown in culture continuously for extended periods of time, sometimes many months. In a complex model such as this, it is important to remove any source of experimental variability and uncertainty. Some of the first products developed in-house at Qkine were those to address the unmet needs of the organoid sector, for high-purity growth factor reagents to replace conditioned media with a defined growth factor cocktail. We continue to build our product range to better address customer and collaborator needs such as this. Our goal is to maximize the success of our customers' science, and the team here are always happy to answer questions and work with you to find solutions to stem cell culture challenges.